Alright guys, so this is going to be an exam review for chapter 3. I'm making this in the last minute and I'm just going with it and doing this right off on the fly actually. So I'm not going to be doing a lot of typing. Pretty much I'm going to be narrating most of this, so be sure you have a paper, a pen handy, and take down lots of notes. Also, please don't email me asking me for answers. I would like you guys to use your books to find the answers for yourself, or use the comments section in the area below. Ask one another, see if you guys can help one another out. So let's start off with this. So on any exam, there's always going to be pictures and diagrams to go over. So two diagrams that I think will probably be on this exam are, this is the first one. So this is that of a cell. So what should you know from here? Well, how about this? What is this structure over here? Okay, what is it called? What is it for? What's the purpose of this structure over here? What are these little things over here? What are they called? What are they for? So make sure you know not only what they are, but what purpose they serve as well. So for example, the question could be, which one of these organelles, okay, remember what organelles are. These are all organelles. Which one of these organelles produces ATP? Okay, so that could be one question. Which one of these guys produces ATP? And of course, the answer would be this, the mitochondria. So another question could be, which one of these organelles is the site for protein synthesis? Okay, another one could be, which one of these organelles is the site for lipid synthesis? And how about which one of these organelles packages proteins? Another question would be, which one of these organelles plays a role in cell division? Which one of these organelles forms the mitotic spindles? Which one of these organelles contains enzymes for autolysis? So this should give you an understanding of the type of questions you're likely to see about this diagram here. So moving on to the next picture that I think will probably be on an exam is the cell wall, or the cell membrane, or the plasma membrane. So again, questions. So again, right over here, remember, you're just looking at the cell wall over here. This is a close-up of it. It's magnified. So questions that you would expect to answer for these type of, from this illustration would be, which one of these structures, or for example, let's just go over here. What is this? Okay, and then you'd have to answer that question. Where are the integrated proteins? All right. Where's the polar region of phospholipids? Identify the polar region of phospholipids. Identify the peripheral proteins. Okay, so we have peripheral proteins, the integral proteins you should know. Again, make sure you're able to identify the polar region of the cell membrane and the hydrophobic region of the cell membrane. So the polar and the non-polar regions of the cell membrane, make sure you're able to identify that. Where's the cell's interior? Where's the exterior of the cell? Make sure you're able to identify that. Where's the glycocalyx? Identify the glycocalyx. So that's probably going to be it for this picture over here. Again, make sure you know what all these structures are. And the second part to it is make sure you know what function they serve. So just don't be able to name them, but make sure you know what they do as well. 2 parts. Next, what I'll be doing is asking you some questions from these set of terms over here, here, and here. So we'll start off with these, this group of terms over here. First question would be, which one of these terms, again, you have to use one of these terms, so it's a matching type of thing, all right? So I'm going to ask you the question, match that term to, match that statement to one of these terms over here. Which one of these may be attached to the endoplasmic reticulum or found scattered in the cytoplasm? Which one of these provides the energy that's needed for synthesis reactions? Okay, which one of these provides energy for synthesis reactions? Which molecule specifies the exact sequence of amino acids of the protein to be made? Which molecule specifies the exact sequence of amino acids of the protein to be made? Next question would be, which one of these attaches the correct amino acid to its tRNA? Which one of these attaches the correct amino acid to its tRNA? Next, which one of these forms part of the subunit for the protein synthesizing organelle? Which one of these forms part of the subunit for the protein synthesizing organelle? And the next one is, which molecule binds to a specific codon and specific amino acid simultaneously? Which molecule binds to a specific codon and specific amino acid simultaneously? So I asked about 6 questions, and there's only 5 terms, so that means one of these you can use twice. Now we'll move down to these set of terms over here. So I'll read a statement. Again, you want to match the statement to the term. And so this goes over mitosis, the phase of mitosis. So in which one of these phases does chromosome uncoil to form chromatin? Which one of these phases do chromosomes uncoil to form chromatin? Next question would be, in which one of these phases does the chromosomal centromeres split and the chromosomes migrate to the opposite ends of the cells? In which one of these phases do chromosomal centromeres split and the chromosomes migrate to the opposite end of the cell? Next question, in which one of these phases does the nuclear membrane and nucleolus disintegrate? In which one of these phases does the nuclear membrane and nucleolus disintegrate? Next question is, in which one of these phases do the chromosomes align on the spindle's equator? In which one of these phases do the chromosomes align on the spindle's equator? Next question would be, in which one of these phases do the centrioles move to the opposite ends of the cell? In which one of these phases do the centrioles move to the opposite ends of the cell? Now we'll move down to these terms over here. So first question would be, which one of these organelles plays a role in the synthesis of steroid-based hormones and proteins? Which one of these organelles plays a role in the synthesis of steroid-based hormones and proteins? Next question, which one of these organelles facilitates peptide bond formation between amino acids? Which one of these amino acids facilitates peptide bond formations between amino acids? Next question, which one of these organelles plays a critical role in the forming spindle fibers for cell division, organizing and moving organelles, as well as giving shape to the cell? Okay. Which one of these organelles plays a critical role in forming spindle fibers for cell division, organize and move organelles, as well as giving shape to the cell? Next question, which one of these organelles are the dense spherical bodies in the nucleus that are the synthesis sites for ribosomal RNA? Which one of these organelles are the dense spherical bodies in the nucleus that are synthesis sites for ribosomal RNA? Last question, which one of these organelles is the vast majority of the cell's genetic material housed? Which one of these organelles is the vast majority of the cell's genetic material housed? Now let's move down to these terms over here. So, which one of these helps prevent molecules from passing through the extracellular space between adjacent cells? Okay, which one of these helps prevent molecules from passing through the extracellular space between adjacent cells? Which one of these is a type of anchoring junction? Which one of these is a type of anchoring junction? Next question, which one of these allows ions and small molecules to pass through from cell to cell? Which one of these allows ions and small molecules to pass through from cell to cell? Next question, which one of these is present in electrically excitable tissue? Which one of these is present in electrically excitable tissue? Next question, which one of these is abundant in tissues that's subjected to great mechanical stress? Which one of these is abundant in tissues that's subjected to great mechanical stress? All right, that's it for matching. Now I'm going to go over some statements, and these will probably be more of a true and false type of scenario. So let's start off with the first question that I'll present to you. Again, I'm not going to type them out, so just listen and write it down. During mitosis, at the end of mitosis, the two cells, do they have the exact same genetic composition? Okay, so at the end of mitosis, do each one of the two daughter cells have the same genetic composition? Next question. What's apoptosis? Or here, let me put it a different way. Is apoptosis programmed cell death? Does apoptosis occur in cancer cells? Next question. Is metaphase, prophase, telophase, and anaphase the correct sequence of order for mitosis? Next question. Is osmosis the passive movement of water? Is osmosis the passive movement of water? Next question. Is the genetic information encoded in DNA the alteration of sugar and phosphate molecules? Is the genetic information encoded in DNA the alteration of sugar and phosphate molecules? Next question. Is DNA transcription another word for DNA replication? Is DNA transcription just another word for DNA replication? Next question. Is the glycocalyx made up of glycolipids, glycoproteins, and cholesterol molecules? Is the glycocalyx made up of glycolipids, glycoproteins, and cholesterol molecules? Next question. Is the glycocalyx displayed on the outside surface of the plasma membrane? Is the glycocalyx displayed on the outside surface of the plasma membrane? Next question. Are microfilaments composed of the protein myosin? Are microfilaments composed of the protein myosin? Next question. Is interstitial fluid found in the extracellular material? Is interstitial fluid found in the extracellular material? Next question. Are aquaporins only found in blood cells and kidney tubules? Are aquaporins only found in red blood cells and kidney tubules? Next question. Are microtubules made up of the protein tubulin? Are microtubules made up of the protein tubulin? Next question. The telomeres, are these the regions of the chromosomes that code for the protein ubiquitin? Telomeres, are these the regions of the chromosome that code for the protein ubiquitin? Next question. Is particle diffusion influenced by temperature and particle size, but not by concentration? Is particle diffusion influenced by temperature and particle size, but not by concentration? Next question. Do concentration differences cause ionic imbalances that polarize the cell membrane and activate the transport process? Do concentration differences cause ionic imbalances that polarize the cell membrane and activate the transport processes? Next question. Which type of RNA has an anticodon that attaches to a specific amino acid? Which type of RNA has an anticodon that attaches to a specific amino acid? Next question. Is the resting membrane potential solely maintained by the passive transport process? Is the resting membrane potential solely maintained by the passive transport process? Next question. Is water able to move through aquaporins? Is water able to move through aquaporins? Next question. Is cytokinesis the division of the cytoplasmic mass into two parts? Is cytokinesis the division of the cytoplasmic mass into two parts? Next question. Is the metabolic or growth phase of the cell's life cycle called interphase? Is the metabolic or growth phase of a cell's life cycle called interphase? Next question. Does aerobic cellular respiration occur in the mitochondria? Does aerobic cellular respiration occur in the mitochondria? Next question. Is sodium the most common extracellular ion? Is sodium the most common extracellular ion? Next question. The process of discharging particles from inside the cell to the outside is called exocytosis. The process of discharging particles from the inside of a cell to the outside is called exocytosis. Next question. In what type of a solution would a red blood cell swell in? Okay. In what type of a solution would a red blood cell start to swell? If a red blood cell were placed into what type of a solution would it start to swell? Would it be a hypertonic solution, an isotonic solution, or a hypotonic solution? Next question. What are two important functions of the Golgi apparatus? What are two important functions of the Golgi apparatus? Next question. If a cell does not have a nucleus, is it going to be able to reproduce? Can a cell without a nucleus reproduce? Explain yourself. Explain your answer. Our next question. Do we only see diffusion and osmosis occur in living tissue, in living organisms? Or does it occur elsewhere too? Okay. Diffusion and osmosis, does it only occur inside the human body or an animal's body? Or does it occur elsewhere too? Does it occur in your kitchen or your bathroom? Next question. What processes maintains a steady resting membrane potential? What processes maintain a steady resting membrane potential? Next question. What's a glycocalyx? What is a glycocalyx? And what are its functions? What's a glycocalyx and what are its functions? Next question. Explain the term genetic code. What is a genetic code? What does it code for? And what are the letters of this code? What are the letters of the genetic code? Next question. Explain hydrostatic and osmotic pressure. Okay, define hydrostatic and osmotic pressure. And the next part of this question is explain how these concepts are applied to the concept of tenacity of the cell. Okay, explain how these two concepts are applied to the tenacity of a cell. Next question. Aside from the nucleus, which other organelle has its own DNA? Okay, aside from the nucleus, which other organelle has its own DNA? Next question. How are the products of free ribosome different from membrane-bound ribosome? Okay, how are the products of free ribosomes different from membrane-bound ribosomes? Next question. What's the difference between lysosomes and peroxisomes? Next question. What's the difference between the G1 and the G2 phase? what's the difference between G1, G2 phase, and what's the difference between the S phase and G0 phase. Okay, so in other words, make sure you know all the subphases. What are they? Be able to explain them. Next question, what are nucleolar organizing regions? What are the nucleolar organizer regions? Nucleolar organizer regions. Next question is, how is a resting potential formed and how is it maintained? How is a resting potential formed and how is it maintained? Next question, list 3 possible causes of aging. List 3 possible causes of aging. Next question, what factors contribute to the fragility of lysosomes and cell autolysis? What factors contribute to the fragility of lysosomes and the cell's autolysis? Next question, what are cell exons and introns? What are cell exons and introns? Next question, why can we say that cells are protein factories? Okay. Why is this statement true, that cells are protein factories? Next, what are lipid rafts? And where are they found and what's their function? What are lipid rafts? What's their functions and where are they found? Next, describes what happens when a protein enters a Golgi apparatus until the time it leaves the Golgi apparatus. Define that process. Next question, compare a gap junction to a channel protein. How are they alike and how are they different? Okay. Compare a gap junction and channel proteins. How are they alike? How are they different? Next question, does phagocytosis occur in macrophages? Does phagocytosis occur in macrophages? Next question, are calcium ions stored in the smooth ER of some muscle cells? Are calcium ions stored in the smooth endoplasmic reticulum of some muscle cells? Next question, is tRNA the RNA responsible for bringing the amino acids to the ribosome for protein formation? Is tRNA the RNA responsible for bringing the amino acids to the ribosome for protein formation? Next question, what would happen to a red blood cell if it were placed in pure water? What would happen to a red blood cell if it were placed in pure water? Next question, describe the plasma membrane. Is a plasma membrane a phospholipid bilayer or is it a double layer of protein that's enclosing the plasma? Next question, would flagella or microvilli, okay, which one of these two, flagella or microvilli, aid the cell in allowing more nutrients to be absorbed? Okay? Would flagella or microvilli aid a cell in allowing more nutrients to be absorbed? Moving along, is it true that the greater the concentration gradient, the faster the rate of diffusion will be? Is it true that the greater the concentration gradient, the faster the rate of diffusion will be? Next question. In tissue such as heart tissue, which undergoes a great deal of mechanical stress, would we expect to see many desmosomes between the cells? In tissue such as heart tissue, which undergoes a great deal of mechanical stress, would we expect to see many desmosomes between the cells? Next question. If cells are placed in a hypertonic solution containing a solute to which the membrane is impermeable, would the cell shrink? Explain why. If cells are placed in a hypertonic solution containing a solute to which the membrane is impermeable, would the cell shrink? Explain why. Next question, what are riboswitches? What are riboswitches? And what is the stimulus that will turn them on or off? What are riboswitches and what is the stimulus that will turn them on or off? Next question, are plasma membrane proteins able to transport molecules through the membrane? Are plasma membrane proteins able to transport molecules through the membrane? Next question, do mRNA, tRNA, and rRNA all play a role in protein synthesis? Do mRNA, tRNA, and rRNA all play a role in protein synthesis? Next question, where in the cell is mRNA found? Where in the cell is mRNA found? Next question, define mitosis. What is mitosis? Define mitosis. Next question, what is a centriole? What's its function? What does it look like? What's a centriole? What's its function and what does it look like? Next question, are microtubules and cilia organelles? Are microtubules and cilia organelles? Next question, what function do glycolipids and glycocalyx serve? What function do glycolipids and glycocalyx serve? Next question, if the nucleotide or base sequence of the DNA strand used as a template for messenger RNA synthesis is A, G, C, G, T, then what would be the sequence of bases in the corresponding mRNA? If the nucleotide or base sequence of the DNA strand used as a template for messenger RNA synthesis is A, G, C, G, T, then what would be the sequence of bases in the corresponding mRNA? Next question, which transport process is the main mechanism for the movement of most macromolecules by body cells? Phagocytosis or receptor-mediated endocytosis? Which transport process is the main mechanism for the movement of most macromolecules by body cells? Phagocytosis or receptor-mediated endocytosis? Next question, briefly explain passive membrane transport. Briefly explain passive membrane transport. Next question, does the smooth endoplasmic reticulum function to produce proteins? Does the smooth ER function to produce proteins? Next question, does the mitochondria contain DNA? If so, what purpose does it serve? Does the mitochondria contain DNA? If so, what purpose does it serve? Next question, are peroxisomes able to detoxify substances by enzymatic action? Are peroxisomes able to detoxify substances by enzymatic action? Next question, what functions do lysosomes serve? What functions do lysosomes serve? Next question, in which stage of mitosis do identical sets of chromosomes uncoil and resume their chromatin form? In which stage of mitosis do identical sets of chromosomes uncoil and resume their chromatin form? Next question, describe the fluid mosaic model of the cell membrane structure. Describe the fluid mosaic model of the cell membrane structure. Next question, what are second messengers? Give 2 examples of them. What are second messengers? And give 2 examples of them. Next question, what's the main component of cytosol? What's the main component of cytosol? Next question, do centrioles organize the mitotic spindle during cell division? Do centrioles organize the mitotic spindle during cell division? Next question, define a gene. Define a gene. What's a gene? Next question, what is crenation? What is crenation? And how could a red blood cell be crenated? How could a red blood cell be crenated? Next questions, are hormones able to enter a cell by receptor-mediated endocytosis? Are hormones able to enter a cell by receptor-mediated endocytosis? Next question, if a tRNA molecule had an AGG anticodon, what would the mRNA codon it would attach to? If a tRNA molecule had an AGG anticodon, what would the mRNA codon it would attach to? Next question, what would happen if a human cell were to increase the amount of cholesterols embedded within its plasma membrane? What would happen if a human cell were to increase the amount of cholesterol embedded within its plasma membrane? Next question, how do our body's white blood cells identify to attack and destroy cancer cells? How do our body's white blood cells identify to attack and destroy cancer cells? Next question, are hormones able to enter a cell by receptor-mediated endocytosis? Are hormones able to enter a cell by receptor-mediated endocytosis? Next question, would decreasing the number of phospholipids in the plasma membrane speed up the rate of diffusion for glucose into a cell? Would decreasing the number of phospholipids in the plasma membrane speed up the rate of diffusion for glucose into a cell? Next question. Does oxygen passively diffuse into our body cells? Does oxygen passively diffuse into our body cells? Next question. Is water moving across the plasma membrane an example of carrier-mediated facilitated diffusion? Is water moving across the plasma membrane an example of carrier-mediated facilitated diffusion? Next question. Would osmosis be limited by low levels of ATP? Would osmosis be limited by low levels of ATP? Next question. How would a cell engulf a large particle? How would a cell engulf a large particle? Next question. If a cell is non-selectively engulfing samples of extracellular fluid, would this be an example of pinocytosis or phagocytosis? If a cell is non-selectively engulfing samples of extracellular fluid, would this be an example of pinocytosis or phagocytosis? Next question. If a cell is selectively reducing the concentration of a particular enzyme in the extracellular fluid, is it likely utilizing receptor-mediated endocytosis or exocytosis? If a cell is selectively reducing the concentration of a particular enzyme in the extracellular fluid, is it likely utilizing receptor-mediated endocytosis or exocytosis? Next question. What's a symporter? What's a symporter? In which type of body cell may they be found? Next question. Would selective diffusion allowing more uncharged particles into a cell assist in establishing a resting membrane potential? Would selective diffusion allowing more uncharged particles into a cell assist in establishing a resting membrane potential? Next question. Describe the function of a cell adhesion molecule. Describe the function of cell adhesion molecules, or CAMs. Next question. Explain why you would expect to find a great deal of mitochondria in a heart cell. Explain why you would expect to find a great deal of mitochondria in heart cells. Next question. Would you expect to see a lot of rough endoplasmic reticulum and Golgi apparatus in the beta cells of pancreas? Would you expect to see a lot of rough endoplasmic reticulum and Golgi apparatus in the beta cells of the pancreas? Next are essay questions. So explain what you would see in the extracellular and intracellular fluid compartments in a severely dehydrated individual. Explain what you would see in the extracellular and intracellular fluid compartments in a severely dehydrated individual. Next question. What does an isotonic fluid loss mean? What does an isotonic fluid loss mean? Next question. What is Tay-Sachs disease? Explain what this is. Next question. In smokers, damage results to the cilia lining the respiratory tract. Explain why these individuals are at a higher risk of developing respiratory infections. In smokers, damage results to the cilia lining the respiratory tracts. Explain why these individuals are at a higher risk of developing respiratory infections. And the last question that I have is, describe the differences between cell division of normal cells versus cancer cells. Describe the difference between cell division of normal cells versus cancer cells. Alright, so that's all I have for Chapter 3 on the cells. I hope you found this helpful. Best of luck to you on the exams. Please be sure to share this with your classmates, your friends. I hope this really helps you. And my one suggestion to you when you're studying for this, there's a lot of material in this chapter. There is a lot of material. There's a lot of memorization. Make sure you look at the pictures, look at the diagrams in this chapter. You have the picture of a cell. If I were you, I would draw my own cell. I don't care what it looks like. Just take a big circle, make a big circle. That could be your cell. Make a mitochondria, okay, and label it mitochondria. Put it in your own words, write it, draw it yourself. Draw all the organelles in it. So draw it as much as you can. This is my advice to you. Whatever photographs, the illustrations that you see in the chapter, try to draw out as many of those as you can and label them on your own. This is an excellent study strategy that I've used in the past, which I found very helpful. Once again, guys, thank you so much and best of luck on your exams.